Hello, everyone. Let's see if I can do this after the intense week. So maybe some of you will be very familiarized with this presentation because we did a very similar one last year, but still, it's a complex topic, it's quite technical, and we have many new families. We have seen so many cute babies joining us in this session. So we were thinking that for the following talks on other therapeutical approaches, it will be really helpful for you to have this one-time overview, also after lunch, that we are all a bit tired. So here we go. If I know how to use this. So CTNNB1 around the world is a network that keeps growing. This is a map that we did from the Spanish organization to use in all our meetings. And very quickly after making this map, it became obsolete. Actually, we still have to update it. Because as you can see here, we have a very extended research network that is framed in gray, and also a tremendous patient organization network that is framed in blue. We have been very lucky to see how this grows with new organizations. We have here Portugal, we have Mexico, we have Canada, and for sure many more to come. The network keeps expanding worldwide, and the new organizations and all the publications, the scientific work, all the efforts that are being done are just a witness of the strength of our community, and seeing you all here is a representation also of this strength. Now, one of those outcomes of these collaborations between organizations has been the CTNNB1, the first CTNNB1 parent organization-led publication. This publication aims to describe, to represent what has been the CTNNB1 community journey, what are the research resources available for scientists all over the world, but also what are the ongoing therapeutical developments. It was already published, and you can see that if anyone wants to consult, anyone is not yet familiarized with it. So my presentation is going to focus in summarizing a bit the information on this paper, and the following presentations will also be around this topic. So first of all, what is the vision of the CTNNB1 community? We are all here in this conference with one very strong topic because, of course, gene therapy is a reality now. But the objective has always been to tackle the disease from different angles with innovative therapeutic approaches, not just maintenance approaches that we do in our daily basis. Ideally, we all dream to develop different therapies for different needs. It can be the severity, the age, the accessibility. And indeed, my presentation has also become a bit obsolete because Damian just presented inclusion and exclusion criteria. So this becomes even more important now that we know that there are inclusion-exclusion criteria in trials, and that maybe some patients cannot be eligible for one therapeutic approach, but they could maybe benefit from another therapeutic approach. So now we are going to see what those approaches are that scientists all over the world are developing for our little ones. So first of all, we're going to have the team here at my front presenting the RNA therapy. How does this therapy work? As I think all of you now know, we have one healthy allele on the gene, and we have one mutated allele on the gene. And the objective of this therapy is to increase the production of beta-catenin of the healthy allele so that this compensates the lack of production of beta-catenin that we have in our mutated allele. If I'm saying something wrong, just please correct me. There are some considerations for every therapy to take into account. Everything has pros and cons. So it is well established for other diseases, already commercialized and used in routine for certain diseases. The dosage is adjustable, and you can do it with certain frequency. It's something that you can put several times in the life of a patient, unlike, for the moment, gene replacement therapy. You can do multiple administrations, but there is a risk of inconsistent delivery and immune reaction leading to side effects. just so you know, this is something very common to basically every therapeutic. So this sentence is going to become a bit repetitive in the next boxes. Next, we will have the gene replacement therapy. This one you're much more familiarized with because this is what we are talking about in this conference. So I will not go so much over the program that we all know very well, but just in the concept of what is done is introducing a healthy copy of the gene into the cells of the patient without touching or doing anything to the genetic material of the patient. We already know that it's well established for other diseases. We have many examples that are helping the development that we have now in our hands. It's a single administration for the moment, and it doesn't integrate in the patient material. The one on the left, the ICV delivered, is the one we are talking about, but there is also another initiative in early stages of development in discovery stage from Tufts University that tackles only the muscle locally. Then when we go into small molecules, I think we have here from the Italian Institute, I'm trying to find also Angelo will talk about it, and also I'm not, if Michelle is online. The goal from the GSK3 inhibitor option is to increase the beta-catenin by avoiding the degradation. So the cells have a destruction complex that regulates the amount of beta-catenin that is inside each cell. If you avoid that complex from destroying beta-catenin, the amount of beta-catenin available in the cell for the necessary functions might be compensated. And I will not talk much about the one of also of Angelo, who will present later, but it is the idea is to enrich locally the pool at the synapses. We had some presentations yesterday that talk about the two functions of beta-catenin. One as a transcription factor, but the other one is where the cells connect to each other. And this is more where the therapeutics of the Italian Institute of Technology are targeting. The good thing of this approach is that it's, again, an adjustable dose frequency, multiple administrations, and it is, again, well established for other diseases. And finally, we would have the DNA therapy, which is the prime editing with CRISPR-Cas system. Maybe many of you have seen in the news very recently, it has revolutionized the field. The case of the first patient-specific approved CRISPR therapy, I think it was on a 6-month-old. They made it in 6 months for a baby in the United States. It was just delivered. So, although indeed it's not yet established, since this document and this was made last year until now, 2 have been approved, one of them for a specific use and one more for general use. So again, this field is a revolution that keeps advancing at giant steps. This technology is different from the others. The others were either promoting the healthy allele or copying a gene next to our genetic material. Or the small molecules are not touching much our genome. In this case, you really do a sort of copy-paste where you try to replace the defect for a healthy copy of in the genetic material of the patient. I'm trying to say it in very general words. As you can see, a few of these options are still in early stages of discovery, but also we have several that are already in preclinical phase. And of course, the ICV delivered that is almost reaching clinical trial. I have no doubts that we will, next edition of this conference, we will be amazed by the advancements that have been done in all of these fields, both from other diseases that we can use as example as of our own. But again, it is the patients, it is the families, it is people like all of you here, it is people like Espela, that are the ones inspiring the research, asking the questions, but also driving this research, driving the development of innovative therapies. And it is the way that they are transforming the dreams into a reality. And for that, I'm tremendously thankful for all of you, for your tireless efforts, support, and this great community that we have created. So thank you all and enjoy the last sessions of the day.